So our last talk of this session is going to be from Jasper Hajonides. You can again correct me on your name. I apologize for that. And his presentation is titled "Decoding Color from Scalp Measured Electroencephalography."
Yeah, thanks a lot for the introduction and for putting this whole conference together and for the invitation to speak. So today I'm going to talk about how we can decode color using EEG. And to make a bit more clear what we're going to be looking at, we're not going to be looking at just decoding green versus red, for example. But in this study, we'd like to investigate whether we can decode a whole range of different colors. So decode color more parametrically using multi-class classifiers. And in the current experiment, we're also going to be looking at multi-item displays. So we're going to be trying to decode multiple features at the same time from a visual display. And importantly, we'd like to look at visual color as opposed to kind of verbal labeling of colors. So we're mostly going to be focusing on the posterior sensory electrodes in our analysis. So before we go into the methods and the results, I would just like to point out that these results have been written up in a preprint, which is available on bioRxiv. We also put out the data on Open Science Framework. So if you would like to have a look at the data, you're more than welcome to. And also put together all the decoding functions in this Python toolbox, which you can download as well and use on your analysis if you choose to do so. So without further ado, I'd like to introduce the task. So the task that we're going to be looking at is a fairly simple one. So here we presented participants with 2 Gabor ratings. Each had an independent color and independent orientation. And what we basically tried to do is to decode all these 4 different features, so the 2 colors and the 2 orientations, in the period after the visual presentation. And at the end of the trial, participants recalled one of the features, either one of the colors or one of the orientations. And these data has been taken from a recently published task, which you can read up if you would like to do so. So we tested 30 participants and 960 trials per participants. Participants were presented with 48 distinct colors, which for our analysis, we binned into 12 different bins to increase the amount of trials per stimulus category. And we used linear discriminant analysis in our decoding. So in conventional decoding, what people usually tend to do is you take the sensors of interest. So in this case, we look at posterior sensors. So you take the 17 posterior sensors from one time point, and you try to train and test your classifier, and you get some evidence measure. And you repeat this process for every single time point until you come at the end of your trial. What we did here is something slightly differently. So we specified a window, and we concatenated all the data from that window into a single vector. So in this case, we'd have a window of 5 time points, resulting in 85 time points. And we slided this window over our data until we've reached the end of our time window. This kind of decreases the noise in the data and allows us to pick up on the difference in temporal dynamics between the stimulus categories. And we also applied a baseline correction within the window. So we demeaned our data within the window to avoid baselining issues. And for the upcoming results, as I mentioned before, we looked at the 17 posterior sensors, and we used a time window of 200 milliseconds. So that basically means that we used 20 time points to do this sliding window analysis. And well, what can we expect in our results? Well, it could be the case that after the visual presentation of the stimulus, our classifier can perfectly predict the presented color. So if we put the target color at the center, it will look something like this top figure. Could also be the case that the classifier is usually correct, but sometimes mixes up the correct stimulus class and the adjacent stimulus categories. And of course, it can also be the case that the classifier has no idea what's going on and just is at chance. So I wouldn't be giving this presentation if the latter was the case. So we actually see reliable tuning of the data towards the stimulus category after the stimulus has been presented, both for color as well as for orientation. We observe these results. And what we can do, we have this 2D tuning curve now, but to better statistically evaluate these results, we can convolve this 2D tuning curve with a cosine functions to obtain this more one-dimensional measure. And yeah, this stresses again that we can reliably decode visual color roughly 100 milliseconds after it has been presented, up until almost a second later. And these results are very similar for orientation as well. So we see very similar pattern for these two visual features. Now you can, of course, think that, yeah, I mean, it's nice that this classifier can pick up on these visual features, but classifiers pick up on all kinds of crazy features. So to get into this in more detail, we ran a searchlight analysis over the entire EEG topography to see where the left and right stimuli are most reliably decoded. And we actually observed that in the contralateral posterior sites, we observed most evidence for these different stimuli, which we would expect based on anatomical organization of the visual system. And we can contrast left versus right to get this contrast even more clear. So this kind of suggests that the posterior visual sensors most strongly drive this effect. And you can also think that this is nice, but maybe this is just driven by one or two colors that can be decoded really reliably. So in order to make this point even more clear, you have to demonstrate that it's not just one or two colors, but we can decode a whole range of these colors. And that is also what we show. We show that all colors can be reliably decoded in these analyses, as well as orientations, although for orientations you have this more cardinal bias that is present in the decoding analysis. And you can take this a step further and you can see how the neural correlates of different colors, how similar they are. So you can create the Dissimilarity Matrix for the neural signatures of all these different colors. And you can then also apply multidimensional scaling on this Dissimilarity Matrix. And what we then observe is that there is this fairly strong circular organization of these colors in these posterior electrodes, as well as for orientation, although for orientation it looks a bit skewed. Now, one of the final results is that even though we defined our colors from a single slice in C-lab space, it can of course be the case that after rendering our colors, that there might be some offsets generated in different features. For example, luminance is a strong one, so it could be the case that there are differences in luminance between the colors that actually drive our results. And so we measured our colors on the screen. We observed that for chromaticity, it's still very circular, but we did observe some very minute differences in the different luminances of the colors. So we wanted to test whether these differences in luminance actually explained our results. So we put together these dissimilarity matrices. So for example, the top left dissimilarity matrix kind of indicates the dark colors show that these colors have very similar luminance, and the lighter colors show they have very more dissimilar luminance. So we want to see how much this pattern is present in the data, and we compared this with kind of the measured colors. So the similarity of the measured colors here expressed as Euclidean distance between the different colors, and we compared this with the model that we used in our previous analyses. And we actually found that we could reliably decode the luminance of the different colors, but with much lower confidence than the actual color properties. And yeah, just finally, we also put together this tool where we subsample the amount of participants and the amount of trials per participant to as an aid for anyone who would like to do something similar in the future to see how much trials or participants they would need, which you can further check out in our preprint. So just to summarize, we show that color decoding reflects the sensory qualities located in posterior electrodes, and it's possible to decode features from multi-item displays. And color decoding has a comparable magnitude to decoding of visual, stimulus orientation, and critically orientation, spatial frequency, motion. They all have spatial components, so they're more confounded by alpha power, eye movements, and visual color would have less issues with these confounds. So it might, in some cases, be a more clear measure that research can use in the future. So I'd just like to thank my supervisors, Mark Stokes, Freek van Ede, and Kia Nobre for their supervision, and all my colleagues in the labs and funders. Thank you very much.
All right, thank you, Jasper. That was a great presentation. I want to get right to the questions because we only have a few minutes here. We've got one comment that just popped up in the chat from Bevel Conway. The MDS plot for color shows a striking bias with end spectral colors, red and blue, forming the poles. Since the end spectrum colors will be consistently differently impacted by chromatic aberration, it raises the possibility that decoding is driven at least in part by individual differences in luminosity settings rather than hue. He does say excellent talk, by the way, which I definitely agree was an excellent talk. But do you want to give any more details about luminosity and hue?
So yeah, that's definitely a good point. And we tried to get at the issue in the final analysis where we show the contributions of luminance and different chromaticities. And of course, in this MDS plot, you can also see that indeed there is some kind of bias with different parts of the color space more clustering together. Although, and this has also been described in previous studies, but yeah, we do show that it's not, I think we try to show that it's not completely driven by these separations, so that it is more a parametric parametric distribution, which can also be seen in individual colors and in the fact that it's actually somewhat circular, that they're not even more clustered together. So it is a concern, but it's probably not.
Yeah, it'd be interesting to try to do some type of variance partitioning to see the extent to which you can explain different amounts of variance between color and hue. It'd be a different analysis approach, I guess, than the one you chose, but it would be interesting to see. So we've got a question from Paul Zur. Do you expect a similar limit in the amount of targets that can be decoded? For example, in orientation decoding, what do you think the maximum amount of color or orientated targets held in memory you would be able to decode? So how many targets do you think it's possible?
Yeah. So at the moment, it will be like 4 distributed across 2 objects. And yeah, I think once you push this a bit further, I think it might damage the classifier fairly substantially, because it probably exceeds the limits of working memory. So in some cases, you might encode one color, and in other cases the other. And then if you train and test your classifier, you might also include training data that does not reliably represent the colors and orientation. So I think 4 features is already pushing it a little bit. I would expect that it does significantly drop off if you include more items, but that would be an interesting future path, I think.
Cool. And then Miguel Santos says, what are the neural mechanisms underlying the color decoding? And why did you choose posterior electrodes?
We chose posterior electrodes because we're mostly interested in the visual aspects, so less so than just a verbal labeling of red, green, blue. And yeah, we also show that it's mainly caused by posterior